we are at an off time from our usual meeting, so that may impact attendance some, though people seem to be filing in to our virtual room. So I want to welcome everyone to a special Tuesday edition of the DMICE Research Conference. Today, we're going to hear from Jennifer Sullivan, who, as most in the audience know, is a PhD student and National Library of Medicine Fellow in our Biomedical Informatics graduate program. She received her bachelor's degree in commerce from Santa Clara University in 2010, and then completed a post-baccalaureate health professions program at UC Berkeley Extension in 2017. She joined our program in July of 2018 as a bioinformatics and computational biomedicine student, and her advisors are Shannon McWeeny and Ronan Schweitzer, who I see is here in the audience. And
also Guanming and Andrew.
Okay, great. Guanming, yes, and Andrew Addy, however you pronounce it, sorry. Anyways, she has an interest in computational methods for understanding rare disorders and using genomics data to understand development of the musculoskeletal system. And so she has worked under their mentorship and focused on using single cell transcriptomics to study the development of tendons. So she is going to give her symposium today, which, as most of you know, is the first step towards working towards a dissertation. And she'll complete this symposium, which gives background on her work as she formulates her research questions for her dissertation. We will hear back from her in the not too distant future for her dissertation proposal defense, and then ultimately her dissertation defense. So I will turn it over to Jennifer.
All right, great. Well, thank you for that introduction. So I'll be presenting on investigating mechanisms of rare disorders through the prediction of disease gene associations. And I would like to begin my presentation by sharing a personal story with all of you. It's not moving. Okay. So this was me shortly after I was born. My birth was normal, with the exception of 2 broken bones and very obvious contractures of all of the joints in my limbs, meaning my joints were very tight and stiff. Because of the contractures and my very thin, delicate bones, I had been whisked away to the neonatal intensive care unit, where I ended up spending the first 10 days of my life. So that's where I am in this picture. Due to physical therapy, I'm actually looking quite a bit better here, but at the time of my birth, I was a tight little bundle, and my parents said that I looked like a little football. Something that you can still see in this picture is this sort of tangled look to my legs, again due to the contractures. After the 10 days in the NICU and multiple incorrect diagnoses, I was sent home with no diagnosis, except for a statement that I had severe arthrogryposis multiplex congenita, which refers to my multiple contractures and is not actually a diagnosis, as I will explain later in my presentation. My parents had no explanation for what had caused my congenital malformation. So far, I had fortunately been able to breathe on my own, as well as smile, cry, and open my eyes, but beyond that, I had made no other movements. My muscles in both my limbs and my trunk were hypotonic and extremely weak. My parents didn't know if I would ever be able to move. My story describes my first days living with a rare disorder. With no diagnosis, my case is an N of 1, unable to be labeled and grouped with anyone else. Even with my designation of having arthrogryposis, I am a member of a small group. Because of the small numbers of people affected by individual rare disorders, most of these disorders have received relatively little attention from the scientific community. Still, although the specific details of my story are unique, the story of someone having a rare disorder is not rare at all. In fact, the truth is that rare disorders are quite common. In this presentation, I'll be talking about a certain class of methods that can be used to understand rare disorders, specifically methods for predicting disease genes. I'll begin by talking about the impact of rare disease and why we should care about developing methods for the rare disease community, despite the small number of individuals with any given disorder. I will also discuss some of the biggest challenges in rare disease research. I will then talk about gene prediction and how it can be used to investigate rare disorders. Finally, I will review the prior work to develop methods in this area, and I will highlight opportunities for the development of new methods. So what is the impact of rare disorders? In the United States, the Orphan Drug Act of 1983 defined a rare disorder as one that affects fewer than 200,000 people in the US. Based on this definition, there are about 7,000 disorders that are considered rare. Within the United States, an estimated 25 to 30 million people have a rare disorder. That's about 1 out of every 10 people. Based on the EU's definition of a rare disorder as one that is life-threatening or chronically debilitating, and that affects fewer than 1 out of every 2,000 people, the global prevalence was estimated in 2019 to be between 263 and 446 million people, or about 3.5 to 6% of the world's population. An estimated 70% of rare disorders begin before birth or during childhood, and another 18% can begin in either children or adults. This means that most rare disorders are lifelong ordeals, and in many cases are fatal early in life. Rare disorders affect people's lives in more ways than simply the physical manifestations. A survey of over 1,000 adults living with a rare disorder revealed that individuals with rare diseases tend to have worse quality of life outcomes than not only the general population, but also those living with chronic conditions that are more common. So why would that be the case? Another study suggested that the very fact that the disorder was rare introduced additional challenges, especially due to the lack of knowledge about the disorder by healthcare professionals. Due to the dearth of research on rare disorders, the so-called diagnostic odyssey that many rare disease patients go through can last many years. Even if a correct diagnosis is determined, there's often little information that can be offered about the mechanism of the disease, let alone any treatment. In my case, although no one knew what had caused my condition, I was fortunate that there were some treatments available to try to reduce my contractures and build up some muscle. Throughout the early years of my childhood, my treatments consisted of physical and occupational therapy, orthotic devices such as splints, casts, and braces, and at least 6 surgeries. Through these treatments, I did gradually gain some strength and flexibility. However, even with these early victories, the effectiveness of these methods was still quite limited. At the age of 3, I was given perhaps my most important prescription, a license to drive. Yes, my first hot rod was pink. I have lived my life unable to walk or even stand, and while I am lucky to be mostly independent throughout the day, I am still fully reliant on assistance for various activities of daily living, such as simply getting in and out of bed. Currently, there are no treatments that directly target the cellular and molecular changes that produced my condition. The startling reality is that over 90% of rare disorders do not have an FDA-approved treatment. But this doesn't have to be the case. With computational methods that can address the challenges of rare disorders, we can begin to understand the biological processes behind them and start to bring down the number of disorders without treatment. So now that we have a better picture of the impact of rare disorders, let's discuss some of the biggest challenges in researching rare disease. The main challenge facing research on individual rare disorders is limited data. When studying a common disorder, a typical computational design might look something like this. A researcher collects some kind of data type from the patients in a large study, such as one or maybe a combination of these omics data types. This data is then associated with the patient's phenotype data or other metadata to train and validate a model that predicts the phenotype or other patient outcome. The model created can then be used either for prediction of unknown outcomes in new patients, or as a way to infer which variables, such as genes, are most likely involved in producing the observed outcomes. However, this type of study may require hundreds to thousands of patients. In reality, the challenge of limited data is not specific to rare disease, so the methods I will talk about can be useful for more common conditions as well. But rare disorders do represent an extreme challenge in this regard, as their much sparser patient populations make recruitment and data collection a much more challenging and prolonged process. In addition to the issue of limited data, there are also biological challenges that must be considered. Rare disorders may sometimes seem relatively simple. It has been estimated that 80% of rare disorders are genetic in origin, and these are mostly believed to be Mendelian, meaning they are caused by just one gene. So a fair question might be, why don't we just treat each disorder individually by editing the mutations in each of these genes? In the long run, this may be something that needs to happen, at least for some disorders. However, this strategy will not be enough. In addition to this approach currently being intractable, there are also a number of other issues that will necessitate a different strategy, one that is focused not on any individual gene, but on the actual disease process itself. To talk about the biological challenges of rare disorders, I will use arthrogryposis multiplex congenita, or what I will simply call arthrogryposis, as a case study. Understanding these challenges will be important for understanding how gene prediction methods can provide new insights into rare diseases. When I told my story, I mentioned that I had been labeled as having arthrogryposis multiplex congenita, but that this was not an actual diagnosis. This is because arthrogryposis is not a single disorder. Rather, it is the medical term used to describe the presence of contractures, or stiff joints, that are present at birth in multiple areas of the body. In other words, it is a phenotype. It turns out that this phenotype is a characteristic of over 400 different disorders. So far, mutations in more than 400 genes have been associated with arthrogryposis. And like most other rare diseases, many of the disorders presenting with arthrogryposis are believed to be caused by just a single gene. But let's discuss some of the biological complexities that must be considered. The first complexity is that for arthrogryposis, as for many rare disorders, a causative mutated gene is not the whole story. The current consensus is that the contractures develop in two phases. In the first phase, a mutation causes fetal akinesia, or reduced fetal movement. In the second phase, it is actually not the initial mutation, but the reduction in movement that has been shown to produce contractures through changes to the tissues in and around the joint. This means that while repairing the mutated gene may be important, it will likely not be enough to restore normal function once the contractures have already formed. The second challenge is that arthrogryposis is not always caused by a mutated gene. In fact, the most common disorder presenting with arthrogryposis, amyoplasia, is sporadic, with no evidence so far of a genetic origin. Consistent with the concept that restricted movement leads to contracture, the evidence suggests that anything that disrupts the development or function of the tissues that are necessary for movement of the fetus can produce arthrogryposis. Along with mutated genes, this includes dozens of environmental factors, which suggest that some cases of arthrogryposis may involve changes to the epigenome rather than the genetic code itself. In addition, diagnostic efforts for patients with arthrogryposis diagnostic efforts for patients with arthrogryposis have focused so far on mutations in the coding regions of the genome, yet many patients remain undiagnosed. If these undiagnosed patients do have a genetic cause, it may be that their mutations lie in non-coding regions, such as regulatory elements. We can see then that the heterogeneous etiology, complex progression, and extensive histopathological changes of arthrogryposis create a complicated picture that requires us to look beyond individual genes. This figure shows biomolecular pathways that are enriched for many of the genes associated with arthrogryposis. Some of these pathways seem to have more obvious connections to movement, and therefore to the generation of arthrogryposis, while others may have significant, though perhaps less straightforward, contributions. This set of genes and pathways represents our existing knowledge of the initial processes leading to arthrogryposis. If we can understand how these genes relate not only to one another, but also to other genes and the pathways that those genes are involved in, we will have a much stronger starting point from which to understand the processes that result in congenital contracture. Beyond arthrogryposis, understanding the genes and pathways involved in other phenotypes and rare disorders will help us to understand their mechanisms as well. Disease gene prediction methods can help us to work out these mechanisms by discovering potential disease genes and suggesting new molecular pathways, and by prioritizing the best candidates for validation and further investigation. Once the mechanism of disease is more fully understood, gene prediction can also help with developing treatments by identifying the genes that may be the most likely drug target candidates. Finally, gene prediction can also assist with shortening patients' diagnostic odyssey by providing evidence for the involvement of genes that would otherwise not be associated with the patient's disorder. I had mentioned that genes are not always the cause of rare disorders. Later in my presentation, I will talk about ways that gene prediction methods could be used to go beyond genes, to also consider regulatory elements and their relationships to genes. So let's talk about gene prediction and why these methods can help us get around the challenges of rare disorders. Just to be clear, when I talk about gene prediction, I am talking about a method that uses information about a disorder or phenotype to predict which genes out of a given set are involved in generating that disorder or phenotype. Typically, the genes are assigned scores corresponding to their likelihood of being implicated, which enables their prioritization for validation and further investigation. A threshold is then used to select the subset of genes considered to be disease-associated. The set of candidate genes is the set that is hypothesized to contain at least one gene involved in the disorder or phenotype. While the whole genome can be used, limiting the candidates based on knowledge about the disorder can improve predictions. A big reason that gene prediction methods can work well for rare disorders is that the disease information that is needed for the model can actually be quite limited. So let's first view these methods through the lens of how much we know about the genes that cause a disorder of interest. To understand this perspective, let's revisit this figure showing the more traditional way of investigating a disorder. Again, for a rare disease, we will likely not have a large cohort of patients. If there is any omics data from any patients with the disorder, it will likely be of just one data type, like whole exome sequencing or maybe RNA-seq. So we likely won't be working with a very large data set. So let's remove these from the picture. Now, I haven't talked about sources of prior knowledge yet, and those will certainly come into play, but to keep things simple for now, let's also remove them from the picture. So what can we use as inputs to our model? Well, that depends on how much we already know about the genes involved in the condition we're investigating. Let's first look at the worst case scenario, where we have no information about any genes associated with the disorder. Here, the goal is to just find any genes that may be linked with this disease. In this scenario, the input to the model could simply be keywords or ontology terms, such as from the human phenotype ontology or the gene ontology, that pertain to the disorder's phenotypes or biological processes. There are multiple approaches that have been developed for this problem, but they all involve predicting genes for one disorder based on its similarity to the phenotypes of other disorders. These include text mining approaches, as well as methods that use disease gene association networks and machine learning. For other disorders, we may have information about genes whose expression is correlated with the disease phenotype. This might look similar to the scenario for more common disorders, where omics data is correlated to patients' phenotypes. Again, though, with rare disorders, we likely don't have the patient numbers or the multiple data types to be able to train and validate supervised machine learning models. Instead, what we have might look more like this. Data from a very small cohort of rare disease patients and controls, and a single data type, such as RNA-seq, producing a small data set. In this case, we can see changes in expression, but it's not obvious which genes are driving the disease state and which ones are passenger genes, more peripheral to the disease process. For this scenario, investigators have used unsupervised clustering algorithms to detect disease gene co-expression modules. Rather than returning a list of individual genes, these methods produce subnetworks of genes that can be further investigated. Finally, due to the increasing availability of sequencing technologies, whole genome and whole exome sequencing, and to a much smaller extent, GWAS, have identified some of the causal genes for many rare disorders. These known disease-associated genes can be used as seed genes to aid in the discovery of additional genes that may be causal or may otherwise be central to the biological mechanism of the disorder. With these models, you don't need to collect any omics data from patients beyond these known disease genes, and this makes these models very appealing for investigating rare disorders. There are a number of different types of methods that use seed genes, including similarity profiling, network-based methods, and machine learning methods performed on networks. One thing to note is that even if no genes have yet been discovered for a disorder, there are still ways to select seed genes to use in the corresponding methods. One strategy is to focus on just one phenotype and identify genes linked to any disorders that present with that specific phenotype. The set of genes involved in arthrogryposis are an example of genes associated with a single phenotype. Another possibility is to infer genes that may be implicated based on their involvement in similar disorders. In fact, many disease gene prediction methods incorporate a disease similarity network and are therefore able to accept either seed genes or a disease term, as we saw before, from which they can infer seed genes from the disease network. Because of this overlap in methods, I will highlight only a few examples when I talk about using keywords to predict disease genes. So let's talk about some of the methods that have been developed. Again, if we don't know any genes associated with our disorder of interest, we can use methods that make predictions based on keywords or ontology terms. Some of these methods mine the literature for associations between phenotypes and genes. In most cases, these relationships are identified by statistically assessing their co-occurrence in published abstracts and papers. For instance, a method called Beagle accepts any keywords, such as phenotypes associated with a disorder, and searches for genes that are mentioned with any of those keywords in Medline abstracts. It assesses the strength of the keyword gene association by calculating the Jacquard similarity, which compares the number of abstracts in which a keyword and gene appear together to the number of abstracts containing just a keyword or just the gene. While text mining methods are limited to genes already documented in the literature, they may reveal new gene-disease associations that can be the starting point for further investigation. The other methods that accept keywords are based on the modular nature of genes, which is the observation that genes that work toward a common function and produce a common phenotype often work together. By looking at disorders that are similar to the one we are investigating, we may be able to identify genes that are implicated in our disorder. Typically, this is done using network-based methods. For example, a method called PRINCE calculates a score for each disorder in the OMIM database based on how similar its mesh terms are to the disorder of interest. So, for instance, if our disorder has a phenotype of arthrogryposis, then another disorder with a phenotype of muscle weakness would get a higher similarity score than a disorder characterized by osteoarthritis, since arthrogryposis and muscle weakness are more closely related in the mesh ontology. These disease similarity scores are then used as seed values for the associated disease genes in a protein-protein interaction network, and PRINCE propagates this information through the network using a diffusion algorithm. More recently, graph learning methods have been used to try to learn the most meaningful relationships in the data. For example, a method called Progeny embeds a heterogeneous network of various biological data types into a lower dimensional representation to try to extract the most meaningful representation of the data. It then uses these refined relationships to construct a new dual-layer disease gene network, which it uses to predict the disease genes. Now let's look at a few examples of methods designed to use expression data that correlates with the disease state. With these methods, we are looking for groups of genes whose co-expression patterns change in the disease samples versus the control samples. By far, the most widely used module detection method is WGCNA, which uses hierarchical clustering to identify modules of co-expressed genes across samples. However, while this method may work well for some applications, it may not be the most effective for zeroing in on groups of genes that are driving a disease process. This is because it identifies co-expression modules across all samples before determining their level of association with the disorder. In contrast, while DiffCoX uses the same hierarchical clustering process as WGCNA, it first takes into consideration the changes in co-expression between disease samples and control samples before identifying modules. Therefore, the modules represent sets of genes that are differentially co-expressed between cases and controls, and therefore likely to be more directly related to the actual disease process. In addition, these methods can be improved by incorporating additional information. For example, Wu and Stein used a protein functional interaction network as evidence of true associations, and they used co-expression data to weight the edges of this network. They then perform Markov clustering on this weighted network to identify co-expression modules. Next, we'll look at examples of methods that can use known disease-associated genes, referred to as seed genes, to inform their predictions. Because seed genes are typically directly related to the mechanism of the disorder or phenotype, the use of seed genes produces the highest confidence predictions of genes that are also involved in the disease process. Therefore, I will spend more time on this category. The first class of methods I will talk about are similarity profiling methods, also known as data fusion or ensemble methods. Genes that perform similar functions or participate together in a pathway will often share similar sequences, have overlapping functional annotations, be co-expressed, or demonstrate similarity in other ways. Similarity profiling methods use these similarity characteristics to statistically determine which candidate genes have the highest resemblance to the seed genes that are input into the model. One of the most commonly used similarity profiling methods, called Endeavor, gathers different types of information about the seed genes by accessing 75 different data sources, providing insights on such characteristics as function, pathway, expression patterns, sequence, and chemical interactions, among others. It then compares each candidate gene to the seed genes and calculates a final aggregated score of similarity. These methods make an assumption that the various pieces of evidence should all have an additive effect on a gene's disease probability. Therefore, to get around this limitation, machine learning models have been developed to learn the contributions of the different evidence sources in predicting disease genes. For example, pBrit extracts information about the genes from various sources and uses this information along with the seed genes to train a Bayesian ridge regression model. The great thing about similarity profiling methods is that they can easily take an unlimited number of data sources into account, providing a wide range of evidence for possible relationships between seed and candidate genes. However, these methods do not explicitly model genes' multiple interactions with one another or consider the complex association patterns between genes and other associated data. Therefore, networks have become a popular structure used to model the actual physical interactions and functional relationships between genes, as well as their associations to other entities. Network-based methods are based on the principle of guilt by association, again referring to the observation that genes operate within functional modules. Early methods were limited to looking at the local environment around seed genes. For example, Diamond works by first identifying the direct neighbors of the seed genes and determining which one is most highly enriched for seed gene connections. That gene is then labeled as another seed gene, and the process is repeated. Although the process expands across the full network, only direct neighbors are considered at each iteration. This assumption that disease genes are directly connected to the seed genes introduces the possibility of false positives and also risks missing genes that are important to the disease process. In response to these limitations, Kohler et al. Introduced what would become very popular methods, the random walk with restart and the diffusion kernel method. These network propagation approaches measure the strength of the relationship between two genes based on the probability of encountering one gene when traversing the network starting at the other gene. These methods can also be thought of as measuring the flow of information through the network starting at a given gene. An example from the original random walk paper demonstrates that two genes separated by another can be scored differently based on the connectedness of the gene between them or based on the number of paths connecting the two genes. Therefore, network propagation methods consider more global topological aspects than methods based on local relationships. When I talked about similarity profiling methods, I said that their advantage was the ability to incorporate many sources of information about the genes. Omics data is inherently noisy, and if we limit our network to just high-confidence curated protein-protein interactions, we will likely be missing a lot of relevant information due to our incomplete knowledge of these interaction networks. In addition, each individual data source contains its own biases, because certain genes, disorders, or other entities will be more well-researched than others. So an important development in the field of disease gene prediction was the extension of network propagation algorithms to knowledge graphs, which incorporate information from multiple data sources. We have seen a couple examples of heterogeneous knowledge graphs that incorporate protein-protein interactions with disease similarity networks. In 2019, Valdeolivas et al. Went even further by extending the random walk with restart algorithm to multiplex heterogeneous networks. They demonstrated their algorithm by using it to predict disease genes from a network that connects a disease similarity network with 3 layers of different types of gene information. Global approaches using these various network propagation methods have become widely used, but are they truly better than the more local approaches using direct neighbors or shortest paths? To answer this question, Shim et al. Tested both a local strategy and a global strategy and demonstrated that actually neither of these approaches consistently outperformed the other in predicting the top disease genes. Instead, they showed that the performance of a given approach depended on the level of connectedness between the actual disease genes. The method that focused on local properties was better at prediction for disorders with highly connected genes, whereas the method focusing on global properties was better at prediction for disorders with genes that were more dispersed throughout a pathway. For some disorders, the implicated genes are partially connected in a pathway and may require a combination strategy. More generally, we don't know what the true patterns of connection are between many of the genes, which prevents us from choosing the most optimum strategy. To try to get the best of both of these approaches, machine learning methods can be used. With machine learning, both local and global topological features can be selected as input to a model, and the model can be trained to weight these features appropriately to obtain the best prediction. For example, Ramadan et al. Developed a random forest model using 13 topological features from their network, some of them local, like degree and shortest path measurements, and some of them global, like random walk measurements. One challenge that the task of disease gene prediction poses to traditional machine learning models is the fact that only positive training samples, referring to the seed genes, are known, whereas the class label for the vast majority of training samples, the candidate genes, is unknown. Therefore, the model has no definite examples of non-disease genes from which to learn. As a result, a special class of machine learning methods that uses strategies for positive unlabeled learning have been applied to this problem as well. For example, Catapult uses a biased support vector machine to predict disease genes based on random walks of different lengths. Since most of the candidate genes are likely not involved in the disease, Catapult trains the model by selecting a random subset of candidate genes to serve as negative examples. The support vector machine is biased because it penalizes false negatives more heavily than false positives, since the examples of true positives are actually known. As an alternative to hand selecting the features to be used in a model, there now exist methods that can automatically learn to represent a network structure as a set of feature vectors. These vectors can then be used for downstream analyses, including as features in a machine learning model. These graph learning methods include network embedding and matrix factorization, as well as deep learning, which I will discuss in the next slide. Earlier, we saw a method that used network embedding to learn meaningful relationships in the data to construct a more refined network. Another example of the use of network embedding is Smudge, which uses a random walk-based embedding model called skipgram to represent each node's environment as a vector. These vectors are then used as the features in a neural network to predict disease genes. While network embedding methods may perform very well in certain cases, the embedding process is unsupervised or semi-supervised, so the topological features that are extracted may or may not be the most relevant features for predicting the genes responsible for a particular disorder. That's where deep learning comes in. Because deep learning uses a supervised strategy to transform the data as well as predictions, it actually learns the features that are most relevant for predicting disease genes, whether these are topological features of the network or attributes of individual genes or other entities. For example, PGCN uses a graph convolutional neural network to simultaneously embed a gene disease association network and make predictions. In this case, predictions of edges between diseases and genes. So that gives an idea of the state of the field. Now I'll go ahead and talk about opportunities for further research and model development. One thing that I touched on and would like to emphasize further is the use of prior knowledge to try to improve gene predictions. We've seen some examples of this, mostly of methods using protein-protein interaction or functional networks, disease similarity, and disease gene associations. The incorporation of prior information is important because this is another key feature that makes disease gene prediction methods work so well for rare disorders. We have seen that the information we get from rare disease patients is typically rather limited, so we need other inputs to our models that can help us to understand the relationships between genes. Incorporating multiple types of prior information has been shown to improve the performance of disease gene prediction methods. For example, when Valdeolivas et al. Tested their random walk with restart method for traversing multiplex heterogeneous networks, they also tried it on simpler networks with just 1 or 2 layers of information, either protein-protein interactions, pathways, co-expression, or combinations of these. Using known disease genes for over 200 disorders, they found that combining all 3 types of gene information produced the highest ranks for the known disease genes. When they also added the layer of disease similarities, the performance was even higher. So, one way to improve disease gene prediction methods could be to incorporate additional sources of information. Fortunately, we are in an era of big data. Even if we don't have large amounts of data for a specific disorder, many consortiums and research groups have established rich resources of data collected from normal, healthy tissues and cells. From these healthy samples, we can learn the normal patterns of interaction for the disease and candidate genes and use this information to make predictions. While different types of data should give us different insights into these processes, some data types appear to still be underutilized for the task of disease gene prediction. Examples include data types from resources like ENCODE that provide insights into the epigenetic regulation of gene expression. Along these lines of improving performance using prior information, you may have noticed that in Valdeolivas' assessment, co-expression data, while helpful, did not seem to have a huge impact on performance, relatively speaking. This is likely because the authors measured co-expression across 32 tissues and 45 cell lines. Mager et al. Demonstrated that by reweighting a protein-protein interaction network according to the co-expression of the specific tissue involved in a disease, they could significantly improve the performance of their method compared to when they used a generic protein-protein interaction network. Therefore, if the tissues involved in a disorder are known, then the expression patterns specific to those tissues could be used to refine the network. The last few years have seen the generation of single-cell data for many healthy tissues at various developmental stages. These data types provide a level of granularity that may allow for the determination of different disease gene networks for specific cell types. Another opportunity involves extending these methods to provide greater insight into the mechanisms of diseases. While the prediction of disease genes can advance our understanding of the disease process by suggesting the involvement of enriched pathways and prioritizing genes for further study, there may be additional insights to be gleaned using the methods that I have discussed. Again, the incorporation of additional data types may help us here. For example, we can incorporate different types of epigenomic data with gene interaction data. From this network, we might be able to tell, for instance, if one of the predicted genes is a transcription factor that binds to an enhancer that regulates other known and predicted disease genes. In other words, we could determine whether there might be patterns of co-regulation based on the connectivity of predicted disease genes to regulatory factors. Being able to discover these sorts of connections should help in interpreting the results of a disease gene prediction method, especially for graph learning methods that are more of a black box in terms of feature selection and the patterns they find in the network. Finally, the last opportunity I will discuss is developing an improved method for dealing with the issue of unlabeled data. In addition to the approach I discussed earlier, another is to try to identify high-confidence non-disease genes using different criteria. Atta et al. Suggest the use of generative adversarial networks to augment this limited set of labeled genes and thereby give the model more labeled examples to work with. In conclusion, we saw that there are hundreds of millions of people living with a rare disorder, and that we need ways to understand disease mechanisms in order to improve diagnoses and develop the first treatment for the vast majority of these disorders. Disease gene prediction methods can give us a good way to study these disorders because they require relatively little data from patients with rare diseases, and because researchers have developed ways to incorporate data from healthy tissues to improve predictions. By improving and applying methods for disease gene prediction, we can start to learn the mechanisms of rare disorders and work toward a world where having a rare disorder finally is rare. I would like to thank the members of my DAC for their guidance, especially in helping me to find a way to follow my passion and contribute to the study of my own rare condition, despite the challenge of having almost no data to work with. I would also like to thank my fellow students for their feedback and support during this process. And thank you to all of you for attending my talk. And with that, I can take any questions.
Great, thank you. I will start off with a question. It's a very interesting talk and very understandable for those of us who are not down in the weeds on various omics. You mentioned about epigenetics. I was actually starting to think about that before you actually mentioned it, but then that raises a larger thought about other factors that might impact expression in the genome, environmental factors, things like that. Of course, there's potentially so many of them that it may be hard to hone in on one, but is any thought to that kind of data that might impact people who have rare disease? I mean, even exposure in utero to their parents, or I mean, the combinatorial explosion is immense, but I'm just curious if you've had any thoughts along that line.
Yeah, no, that's definitely an interesting route to think about. Again, I think it'll depend on whether that data is available for rare disorders. So for some it may be, for others it may not be. But yeah, no, it's definitely an interesting thought. Thank you for that. Yeah. Um,
Great. Any other questions? You can either type them in the chat or unmute yourself and
This is Shannon. I have one. Really nice talk, Jennifer. Really well put together. I think the thing I was thinking about as you went through the full presentation is, as you mentioned, often there may be one gene and there's other pieces like what actually happened developmentally. And so it seems like depending on the phase you're interested, are you interested in like, prenatal diagnostics? Are you interested in identification of disease gene when it's not known? Are you interested in therapeutic options where you're now thinking about trying to ameliorate certain effects, which may, resulted from the disease gene, but may actually now be quite downstream? And so I was just wondering, have you thought about where you want to focus on that? Because these methods obviously can be applied in a lot of different ways, and some, depending on the question, for example, if you're really truly mechanistic, you're thinking much more about causal inference than you are about association. So I was just curious where your thoughts were leaning in terms of direction for your dissertation.
Yeah, exactly. I guess that's kind of why my focus, that I put my, more focus on the section of using the seed genes to kind of understand genes that are sort of directly related to the mechanism of the disorder, hopefully. Yeah, so I'm like in my personal case, since I'm interested in arthrogryposis, it's kind of an interesting question because, like I said, the initial mutation is not actually what causes the contractures. The contractures develop after movement stops. But I think if you're going to use seed genes, like what we have for arthrogryposis, for instance, is those like initial mutated genes. And so like for me, I think it would be, helpful to understand just even those initial processes, what causes movement to stop in the first place. Or, at least the initial parts of that. It'll be, somewhat limited in terms of how far along the process we can go until we kind of, understand those initial processes, and that's kind of what we have the data for right now, if that makes sense. I don't know if I explained that well.
That's great, thanks.
So Jennifer, this is Ronan.
Hey.
So I wanted to ask you about specifically about arthrogryposis, since unlike many of the other conditions that you're talking about, here you have an embarrassment of genes, right? You're looking at a situation with a huge number of genes that have been associated, and I wonder if similar approaches can be used within this group of genes to try and see if there are analytical ways to establish rank, significance, or interactions between these, or can these things be used can these approaches be used to further our understanding of mechanisms within such a large set of defined genes?
Yeah, no, definitely. I mean, if your goal is to just prioritize the genes that are already there, you can definitely use one of these methods or develop a method that, for disease gene prioritization, and just kind of use those genes that are known as the candidate genes. You might, I mean, still have to have something to kind of, if you're using the seed gene method, you'd have to still use some of the genes as seed genes. So you might choose maybe a subset of those to use, and then try to predict or prioritize the other genes. So yeah, it's an interesting question. But yeah, typically, like when you're doing validation, for instance, on these methods, you would kind of hold out a set of the genes that are known to be involved in a disorder, and then kind of see if those genes rank highly in the list. But that's more for validation. But I think you could certainly do it, something similar with actually using these methods. Does that answer that?
Yeah, I think there's a real question there of defining a goal for what
Yeah.
you're trying to get out of the analysis.
Great. There's a question from Aaron in the chat. Thanks for a nice talk. Could you talk a little about using the vast amount of genomic data available for patients that don't have the rare disease of interest?
Right. Okay. Yeah. So like. Okay. Yeah, that's an interesting question. Yeah. So I mean, I think in that case, you would use a disease similarity network, and then you could maybe, like you would have some sort of protein-protein interaction network, and you would have those like disease gene connections. And when you kind of either traverse the network or learn the properties of the network and feed that into a model, hopefully those, connections to genes or whatever omics data you're using for the related diseases, will come out as important features when kind of predicting the disease genes for your own disease. So that's kind of how those methods work. I don't know if I'm answering your question, if you want to clarify.
Yeah, somewhat. I was just, can you hear me?
Yeah.
I was just thinking that, you don't have a whole lot of information on patients with the disease, but you have a huge amount of information on other patients, either patients with a completely unrelated disease where, you'd expect, that to serve as kind of a negative because the, expression and interaction and other things are very unlikely to be related to the disease, so sort of a contrast. Or maybe even, diseases that target the same tissue or maybe share some sort of phenotype or, you know, which might have some similarity, and being able to contrast those two things in analysis and machine learning. You know, you don't have a lot of positive cases, but you have a lot of it shouldn't look like this kind of thing. So, you know, what's different enough to be helpful in that way? So I don't know. The thought was just that
Yeah.
Judicious and thoughtful selection of a large amount of negative data might be useful.
Yeah, definitely. Actually, that's a really good point, because it goes to the issue that I talked about of having so much unlabeled data. The candidate genes are unlabeled, and so you know what some people have tried is exactly what you said. Look at genes that are associated with totally unrelated disorders and try to use those genes as negative examples. Basically, label those genes as negatives, and then have your model learn from that or whatever you're trying to do. And so, and I know there's one paper that, kind of had like different categories of like, okay, these are like high confidence negatives, and then these are, somewhat confident negatives and that sort of thing. So yeah, that's a good idea.
Well, yeah, I'm actually going to a little bit more, you know, upstream from that. We're not
Okay.
even, if you have the, you know, genomic data on labeled patients,
Mhm.
you don't have to label the genes as negative. You can have the system label the genes as negative in contrast to the few positive patients. So you can be even more upstream and look at those expression of those negative patients, or, you know, other sorts of things, so that you don't have to necessarily make a binary decision at the gene level. You can make it more at the sample level or the patient data level. That was the thought anyway.
Yeah. Okay. Cool. Thank you.
Thanks.
Great. We can probably take one more question. Or if not, we're
Hey,
actually
Jennifer.
oh, go ahead.
I have a quick one. This
Sure.
is Nate. This is kind of a half-formed thought, but I really like I loved your presentation, and I like this
Thank you.
idea of using seed genes in protein-protein interaction networks. One thought I had was we know we have kind of greater or less coverage in certain areas of the proteome, and so the assumption with these seed genes is that the right protein-protein interactions are known. Do you think that there's maybe some work to be done, or a role in adding to those low coverage regions to help maybe understand more of the rare disease interactions?
Yeah, definitely. So I'm assuming you're talking computationally, so like maybe using some kind of imputation or something like that to fill in some gaps, possibly.
Yeah, that'd be one thought.
Yeah, no, it's an interesting idea. I mean, obviously with that you want to be careful not to introduce sort of spurious connections that don't actually exist, but yeah, it could be something interesting to look into. Definitely.
Great. Well, we will look forward to your coming back with your proposal and defending it. And this sounds like some really potentially exciting work. Some of us will look forward to hearing you present at the NLM trainees meeting in a few months. So good. Well, thank you everyone for coming today, and see you all later. Take care. Bye-bye.
Thank you.